So we are now on YouTube at the same time. Good. Just to be sure that's, so we are now on voilà. Otherwise I have a terrible echo. Very good. Good. Good afternoon, good morning to everybody. We have our last committee of expertise this year, and we look forward to spending with you a couple of minutes on a burning topic. Actually, it is using IDMP in adverse event reporting and ICSR. I'm going to introduce the speakers in a couple of minutes, but this is the scene where we play UNICOM, IDMP, and an exchange with the audience through the chat, the Q&A facility, which I'm going to explain you in a minute or so. We have some rules for those who are not familiar with the community of expertise. It's very easy. You are mute. You cannot unmute yourself normally. You're on mute. You are using the Q&A facility. I'm going to show you that on the next slide. If I give you the microphone, please be concise so that we can have an exchange and not have I would say a long statement from somebody. The purpose when we have this dialogue at the end of the presentations is to have an exchange, really. And if you are happy with something, happy with the question also, please use the possibilities you have to express yourself with this button. The way to activate the Q&A facility is first to move a bit your mouse on the screen, and then you see the bar. On this picture, it is on the bottom of the screen. You have a Q&A there, and once you click on it, you see such a window opening. So you see Robert Stegway did ask a question at the very beginning, and then I answered something, and he could comment that again, etc. So you can have a kind of a dialogue with the Q&A. And I said you can also show your approval for a question, which is with the thumb up here. And that's if there are many questions, that brings the approved question a bit higher on the pile. What we are going to do is also to answer the questions at the end of the presentations, as I said before, so that we can have an exchange between the speaker and the panelists. And the webinar is safe. You know, this is something I'll repeat each time because we had some, there were reported some issues at the beginning of the use of Zoom, but nowadays it is very stable and safe. That means that you have received a personal invitation once you're registered, and you are supposed not to share that invitation. So you exist only once on the webinar. We are recording this live. It is currently live on YouTube. Last time we had a community of expertise, we had a bit more than 10 people on YouTube during the webinar. That was due to company policies which did not allow to connect on Zoom. So you see we have this dual possibility. Of course, the dialogue with YouTube is a bit more tricky. Who are the person you are meeting this afternoon? I didn't bring them in a special order, but we have three speakers. First, Anja van Haren from Netherlands. She works at CBG, so the Medicine Evaluation Board in Netherlands. She's a pharmacovigilance specialist there. and she's very much engaged also in UNICOM, where she is one of the lead authors of a deliverable they are going to speak about. Barry Hammonds had a long career in the industry and is a consultant for a couple of years. Anja and Barry are both members of ISO TC215 and Working Group 6. Barry is, in the meantime, also a specialist with pharmacovigilance, of course, and MedDRA, which he represents in UNICOM. Sarah Vaughan is working at MedDRA, and she is also a pharmacovigilance specialist. She's actually a systems manager, and she's completing our trio of speakers. And they are going to have a single presentation where they exchange the speech between themselves. To assess somehow and to, I would say, encourage the dialogue, we have two panelists. From the left, you have Julie James. Julie James with Blue Wave Informatics. She's an HL7 specialist for ages, for long. She's very young still, but she has a long track record in HL7, and she has also a very long track record with IDMP, doubtless. She's also very much engaged in UNICOM in the same work package where Anja van Haren is also working. Jane Miller is representing SNOMED CT here. She's also engaged in UNICOM in the work package 1, but in other work packages as well. And Jane is certainly one of the SNOMED CT specialists we have at the moment. And they will so enrich the dialogue at the end of the presentations. That's the purpose. As I said, you use, please, this Q&A facility. Don't be shy. It's important that you post your question. And again, we will answer them at the end of the presentations. And if perhaps something could not be answered, we will follow up after. If you put if your name is visible, we can reach out to you afterwards. After this webinar, you're going to receive an email asking you to fill a feedback form. Already here you have the link on it. You will find the same link at the end of the presentations as well. We really need a feedback from you. Sometimes people are reluctant to spend a few minutes, and honestly, it's very short. It's mostly to understand your interests about what has been said and try to improve for the next time. With this, I'm going to hand over to Anja, and I will disappear from your screen.
Okay, thanks, Christian, for the introduction and for having us in the community of expertise. We are very happy to introduce to you one of our deliverables from task 8.4. That I'm happy to lead together with Julie. And today is about our guidance that we wrote for using IDMP identifiers in the individual case safety report in the ICSR. So can you move to the next slide, please, Christian? Yes. And in this task, I had the pleasure of working with a very nice team, with nice people, with different expertise. We had colleagues from regulatory agencies, from the UK, from the MHRA, but also from Croatia, from HALMET, and of course from the MEB, the organization I work for. And we also had expertise from coding and terminology with Barry, Jane, and Julie. So we had a very nice team with nice meetings, good discussions that are reflected in the guidance document that we wrote. Next slide, please. So this is just a list with the acronyms. As you know, we regulators use a lot of acronyms. These are the ones that you will see throughout the presentation, and I'm hoping that most of you will be familiar with these. So this is just here for your reference. Next slide, please. So this is what we are going to do today. I will kick off and talk to you about pharmacovigilance as driver for IDMP. It is often said that IDMP is needed for pharmacovigilance purposes. Pharmacovigilance was the driver, but we go back to the very initial use case within the pharmacovigilance area toolkit to give you a better view on that. Also, I will take you to the steps we took to develop the IDMP coding guidance for ICSRs. And then I will hand over to Sarah, and we will dive in a little bit deeper into the guidance itself, and she will take us through some of the principles that we wrote for selecting IDMP identifiers for the purpose of the ICSR. And then we hand over to Barry, and based on the recommendations we have in our document, but also the internal discussions that we had, we have selected some points for discussion. Next slide, please. So when we talk about pharmacovigilance as the driver for developing IDMP, I would like to start with going back to the basics. So the pharmacovigilance definition, as defined by the WHO, is the science and activities relating to the detection, assessment, understanding, as well as prevention of adverse effects or any other drug-related problem. So when you look at this definition, you can distinguish two sides of activities here. First, the activities related to data collection and assessment of suspected adverse reactions. And these activities are carried out by the medicines regulatory authorities, pharmacovigilance centers, as well as pharmaceutical industry. But there are also activities, of course, on the clinical side. This is where healthcare professionals and patients need to be able to recognize adverse effects and take appropriate action to minimize harm. So the outputs of pharmacovigilance analysis will be communicated to healthcare professionals and patients as applicable, for example, by updating patient information leaflets, issuing warnings, things like that. In reverse, healthcare professionals and patients can contribute to pharmacovigilance knowledge by informing industry and regulators about any suspected side effects. So throughout the life cycle of the product, the benefit risk is being monitored all the time via various pharmacovigilance activities. But when it's said that pharmacovigilance was the main driver for developing IDMP, this actually refers to the activities that are shown on the left. Next slide, please. So if you focus on what's happening on the left-hand side, here in pharmacovigilance, data is collected from various sources, such as clinical studies, observational studies, scientific literature. But for the purpose of this presentation and our guidance, we have limited ourselves on a traditional source of pharmacovigilance. And these are, yes, the reports of the suspected adverse reactions that were initially collected through patients and healthcare professionals, and then exchanged between medicines regulators and pharmaceutical industry by using this ICSR message. So ICSR is the acronym for an individual case safety report, and the EU legal definition is that it's the format and content for the reporting of one or more several suspected adverse reactions to a medicinal product that occur in a single patient at a specific point in time. So the international exchange of ICSRs for pharmacovigilance was a key driver for the international initiative that initially started in ICH on the identification of medicinal products. And there were three goals here. First of all, we needed to improve the efficiency of ICSR processing. Importantly, we also wanted to improve the accuracy of data analysis of ICSRs. And of course, since it's a global effort, we also wanted to improve the collation of data globally, facilitating earlier signal detection. Next slide, please. So if I go through this picture, you will get a better understanding why the IDMP is so important for us. We start at the bottom of the picture. This is the flow of how information on suspected adverse drug reactions is running. So we start at the bottom with the patient consumer who is experiencing an adverse drug reaction, and they can report themselves to a national competent authority or a pharmacovigilance center or to pharmaceutical industry. Or if they contact the healthcare professional can contact the competent authority, the pharmacovigilance center or pharmaceutical industry. Anyway, the competent authorities and the pharmaceutical industry have an obligation to report suspected adverse drug reactions to the EMA, to the Vigilance database. Also, the EMA makes available to the WHO Uppsala Monitoring Center the cases coming from the European Union. And what you see here in the blue boxes is that all these organizations use their own dictionary for indicating which drug is involved in the ICSR. So industry often use their own dictionary. The regulators may use an in-house dictionary that's used for regulatory purposes. Some use the WHO drug dictionary, or some use a dictionary that's even used in the clinical domain. For example, our pharmacovigilance database that we have in the Netherlands uses the Z-index. So EMA is using the XEVMPD, the Vigilance Medicinal Product Dictionary, and WHO is also using their own dictionary. And last but not least, if you press the button, Christian, you will see that industry also has an obligation to report the EU cases to other authorities worldwide. And these authorities may even use also their own local dictionaries. So what is happening here is that all these organizations, upon receipt of an ICSR, they are sort of recoding or classifying the information to match it with their own dictionary. So you can see that having IDMP identifiers that we can use within the ICSR will have some efficiency gains here. Next slide, please. So for the exchange of ICSR, we use the ISO standard. There's an ISO ICSR standard, and ICH has written an implementation guide for this. We refer to this as ICH E2B R3. And this format is widely used by the regulators, pharmacovigilance centers, as well as pharmaceutical industry. Actually, this format will become mandatory in the EU for any exchange between the regulators and pharmaceutical industry June 2022. And the ICSR has placeholders for transmitting IDMP identifiers. And within that ICSR, different levels can be used. We always have a free text for indicating which drug is involved, but depending on the information available, we can also provide MPID, PhPIDs, or substance IDs. So use of these identifiers will improve efficiency of ICSR processing as well as data analysis, but selecting these identifiers will have to be done accurately and consistently. Next slide, please. So here we have our famous IDMP wedding cake again, and just to show to you that 2 of the IDMP standards are already almost in use. We already use units of measurement. That's part of the ICSR message anyway. And as from June 2022, we will start using EDQM dose forms and routes of administration in the ICSR. We will use the EDQM terms for that. But when it comes to the other identifiers, the MPID, the PhPID, and the substance ID, of course, we are still waiting for the identifiers to become available. Next slide, please. So I already said that this coding needed to be done accurately and consistently. So we felt that IDMP coding guidance would be needed for the ICSRs, and we will talk you through the steps we took to write this guidance. Next slide, please. So first of all, we contacted primary receivers of suspected adverse drug reactions, pharmaceutical companies, MAHs, as well as the national competent authorities. And we asked them for the drug verbatims. So, and with drug verbatim, we mean what the reporter, so the healthcare professional or patient, has literally stated about the drug when he reported the suspected adverse drug reaction. We also collected data from what we have called secondary receivers, data from the EMA and WHO, what they receive in the ICSRs that have been transmitted to them. We made sure that the data, the drug verbatims that were received came from different reporting media, from web forms or mobile apps or paper forms, phone, or even clinical systems, electronic health records. And we have done this because the drug verbatim is often quite imprecise. There can be a difference if somebody is just calling you about the suspected ADR and just gives you a brand, or when you receive rather precise information from directly from a clinical system. Next slide, please. So we took a sample of 115 of those drug verbatims as provided by the initial reporter, and we coded this with MPIDs or PhPIDs, also taking into account if name parts could be coded. And we did this in line with the guidance that's already available in the ICHE to be R3 implementation guide and in the EU ICSR implementation guide. So just to be clear, there already is a bit of guidance on how to use these IDMP identifiers in the ICSR. And what you see here in the flow on the right is taken from the EU ICSR implementation guide. And it shows you that you will always give free text, let's say drug verbatim, the medicinal product name as reported by the primary source. But on top of that, you will always have to give the most precise identifier that can be selected, coming from MPID, PhPID, or substance ID. So 5 reviewers coded a set of 60 terms, and all these terms were coded by 2 reviewers who were blinded for each other for the paired reviewer. And for each of those drug verbatims, it was considered if the MPID or PhPID could be selected based on the exact drug verbatim, or whether it was possible to assign a more specific PhPID or MPID by imputing information based on some assumptions. And if some assumptions were made, these were also specified. So with that exercise, we identified some areas where we thought further guidance was needed. And this was based on differences in coding results. And interestingly enough, based on the guidance that we have available now in the implementation guides, full agreement between the two coders could only be reached in about 15%, so 15%. Partial agreement in 46%. And in 39% of the coding, there was no agreement at all. Also, we took upon some of the challenges that were raised by the reviewers themselves. Next slide, please. Also, we had to make some assumptions. As you are all aware, generating PhPIDs and what is exactly needed to be detailed in the PhPID is still under discussion. So also, for example, how to express dose form and strength. So different proposals for PhPID generation will result in a more or less granular PhPID. So for our guidance, we assumed that PhPID sets would be available at least for the substance base level, but also for the substance with salt level. Next slide, please. And we made this assumption also because, remember what I said, that this drug verbatim can be a bit imprecise. We have to take into account different levels of precision when data is being collected, based on the reporting source, but also the reporting mechanism. We were thinking that a patient who is reporting via a web form, for example, may use the brand. But if you receive an ICSR or a suspected adverse drug reaction from an EHR system, you may be well aware of which substance was administered in detail. Anyway, we may collect more precise or less precise information. In the end, when it comes to data analysis, we have to make sense out of this by making proper groupings again. Next slide, please. And this is just an example that explains the same principle, that there can be differences in the drug verbatim that is coming to us and how this translates to different level of PhPIDs. Next slide, please. So before we move into the details of the guidance itself with Sarah, we also wanted to give you a little bit of a disclaimer. We wrote this guidance within the UNICOM project. And as you are maybe aware, there is only limited participation from pharmaceutical industry. So industry was not involved in our guidance. We also want to point out that this guidance, it cannot be considered as worldwide agreed regulatory guidance. In pharmacovigilance, our world is really highly regulated. It's also worldwide, and we need to have agreement at the level of ICH before we really can start using the MPIDs, PhPIDs, and substance IDs in the ICSR. We need all stakeholders globally to have a common implementation and understanding. So to best support pharmacovigilance stakeholders, we recommend to explore that another organization like ICH or WHO takes responsibility for owning IDMP ICSR coding guidance and to take this forward when IDMP implementation progresses. And having said this, next slide, please. I will hand over to Sarah, who will discuss with us the principles and guidance for selecting IDMP identifiers as we have drafted in our guidance. Sarah, over to you.
Thank you, Anja, and good afternoon, everyone. So as Anja said, I'm going to be talking through the principles that we've written for selecting IDMP identifiers and use some of the practical examples given in the guidance document to hopefully bring the guidance to life a little bit and help envisage how IDMP can be used in ICSRs, as Anja said, to really promote efficiency in coding and importantly, bring consistency across the different parties involved in pharmacovigilance. Next slide, please. So on this slide, you can see we've developed a summary flow diagram for selecting a MPID or a PhPID based on the drug verbatim in an ICSR, with the aim being to select the most precise term possible. It's quite a small diagram on this slide, so I will talk you through each of those 4 steps. The first step is to check the data field for the verbatim drug information as it's been provided by the reporter and make sure that you have a valid drug in there. Often at the MHRA, we might get reported a class of drugs, or we might get a reporter telling us about a statin, in which case we need to go back to the reporter and seek clarification directly from them about the suspect drug. So once you've done that, the second step is to review the available contextual information to help impute a more precise selection of MPID or PhPID. And over the course of the next few slides, I'll talk through some examples of what sort of reliable information might be available in the ICSR to help support the term selection. And it's important to note that this idea of imputing drug information is a different approach to perhaps what you're used to in terms of coding of reactions using MedDRA, where you have the points to consider for term selection guide, which talks about only coding as accurately as possible against the verbatim and not adding any information or inferring any information. So this is a slightly different approach here for how to get more precise information relating to IDMP. Step 3 is you'll want to consider whether you should be using a current or a historic ID. And I'll go into a little bit more detail on that on another slide. And then lastly, step 4 is you should be selecting the MPID or PhPID, taking into consideration the additional factors you've reviewed in the ICSR. And you're looking to code the most specific term. So if not possible to code an MPID or the PhPID, or a substance ID, then only the drug name as reported would be provided in your ICSR. Next slide, please. So as Anja mentioned, there's often wide variation in how the drug verbatim field is populated. So it can just be free text information or where people type into a web form. It could be use of a non-standardized term list, and this will vary according to the report source. If we're adhering as closely as possible to the drug verbatim, then it's likely there'll be a loss of precision in the term that can be selected if we're not taking into consideration other relevant information that gives some context on the scenario around the drug being reported and gives us some more clues as to what we can code that to. So if you look on the left-hand side of the slide, you can see here are some examples of reliable contextual information, the country where the report was received, the reporting mechanism for how it was obtained, information relating to timing and method of drug administration is useful, and also who the primary receiver is, so whether it's a national competent authority or a marketing authorization holder. And all of this information can help select the most appropriate MPID or PhPID. Next slide, please. So to demonstrate that, we're going to go into some examples now, looking at those other sources of information that can help you to impute the drug information. So in this first example here, we have a suspected adverse drug reaction that's been reported to the Dutch Pharmacovigilance Centre, and the drug verbatim is 0.9 NaCl, sodium chloride. On the basis of the drug verbatim alone, you'd be able to select PhPID level 1 relating to the substance. However, as the report originates from the Netherlands, then the Pharmacovigilance Centre will know there is only one sodium chloride product on the market, and this is a Baxter product. And using this contextual information, the receiver can select a more precise level of PhPID, or even potentially the MPID. Next slide, please. In this second example, we can consider the reporting mechanism as an important piece of contextual information. And Anja has already alluded to at the beginning that many countries have direct integration of ADR reporting from clinical systems, with data coming directly from the patient's medical record, and the term selection used in the drug verbatim coming from a national standardized dictionary of medicinal products. So now this is clearly a very reliable source of information and will enable much greater precision in a term selection that's possible. Next slide, please. Another sort of useful source of context is information around how the drug was received, so the timing and the administration of the suspect drug. And in this example here, we've got an example where it was reported as the suspect drug was named as COVID vaccine. So it doesn't give the name of the vaccine. Fortunately, we know a lot about the timings and the use of COVID vaccinations. And at the time of vaccination in that country, there was only one authorized vaccine available. And therefore, PhPID level 4 could be reliably imputed, and potentially even the MPID if only one was available. If we didn't impute this information, it wouldn't even be possible to code to level 1 of PhPID for this case. So it's a good example of showing you where imputed information is important. And then our last example here around looking at the context around how you can interpret that drug information is about whether it's who the primary receiver is. So in this example here, we have a case where a patient has reported to a pharmaceutical company, and it's an ADR concerning methylphenidate 18 milligrams. Now, because this was reported directly to the marketing authorization holder, and it's about the suspect medication, they can assume it relates to their own product. So that for Janssen, that would be Concerta 18 milligram tablets. And again, that now allows coding at level 4 PhPID. If this had been received by a regulatory authority, it wouldn't have been possible to impute this, and the ICSR would have had to be coded to only level 2, because we would have only known the active substance and the strength. Next slide, please. So if we move on to step 3, then, it's important to note that new MPIDs can be assigned for a product following a substantial change. So that could be a change of active substance, or it could be more administrative, such as a change of the marketing authorization holder. And when you're reporting in ICSRs, there are scenarios where a historic term may be required. So for example, with retrospective ADR reporting, if there is media interest in articles about a particular drug and reaction, then you might find retrospective reporting happening many years later. So for example, hormonal pregnancy tests used in the 1960s linked to long-term developmental defects. You would need a historic identifier in that scenario. Another area where you might need to use a historic identifier is with regards to relevant past drug history. And the example on the slide here is the seasonal flu vaccines, where the ingredients can change year on year, depending on the strains of flu circulating. And again, this is different from what many reporters might be used to for MedDRA coding, where the older terms are no longer considered to be relevant, they're marked as non-current, and they shouldn't be used. This is different in IDMP, where you have this idea of historic terms and current ones, and needing to consider which one might be relevant to different areas within your ICSR when you're coding drugs. Next slide, please. So lastly, then, you've got step 4, and this is the MPID and the PhPID term selection. So obviously, the MPID is the most specific term available that you could use in the ICSR. However, given the specificity of them and that there can be variation in an MPID for a product, it's likely that to start with, coding to MPIDs will be rare. And if more than one MPID is available for a product, then you should be instead selecting the most precise PhPID level. So as a general rule, the MPID can be selected as long as the product only has one MPID and the drug verbatim is sufficiently clear and detailed enough to enable you to impute that, or that you have sufficient details that can be imputed from the contextual information provided in the ICSR. And our guidance document has lots of points to consider when selecting MPIDs or PhPIDs. And you can see on this slide all the different scenarios that we have looked at as a group and written down some guidance around how they should be dealt with. I'm not planning on going into each one of those, but there were just a couple that I wanted to talk through in our presentation today. So if we can move on to the next slide. The first one of these is how to handle brand names with different compositions. So if we look here, you can see the drug verbatim is reported as a brand name, but really this is a brand that refers to a range of products. So for example, in the UK, you have Lemsip. It's a cough and cold remedy. It's available in many different formulations and with combinations of different ingredients. And that's the same as the example here. You can see that the brand is Voltaren, and it has 3 different presentations. We've got a tablet, a gel, and a solution for injection. And the active ingredient is 3 different diclofenac salts. And as Anja mentioned at the beginning, there's still a lot of discussion being had around how PhPIDs are going to be generated, how to express dose form and strength. But we have made the assumption in this guidance document that there will be different sets of PhPIDs for each of the bases and the salts. So a different PhPID for diclofenac sodium and a different one for diclofenac calcium. So in this scenario on the slide here, it wouldn't be possible to code unambiguously if all we had from the drug verbatim was Voltaren. And therefore, we'd only be able to code to PhPID level 1 for the base diclofenac. Next slide, please. So the last example that I wanted to give was around the use of name parts in the ICSR alongside PhPID term selection. And the example you can see here again is related to the COVID-19 vaccines. The drug verbatim is the COVID-19 mRNA vaccine. And so this could be referring to MedDRA or Pfizer vaccine. And we don't know that from the drug verbatim alone, but we do have a lot of reliable contextual information available to help us impute the drug information. So we know the country the drug was received in is the Netherlands and was received from a vaccination registry, and the primary receiver organisation is the marketing authorisation holder MedDRA. So we can therefore reliably impute the drug to be the Spikevax vaccine, dispersion for injection. So if we code to the highest level of the PhPID, because we know the strength and the form and the substance, we would still be losing information around the brand name. And that's why we need to use the name parts data elements. We need to enter the invented names, Spikevax, and form as dispersion for injection. We need to put that in the form side. And we need to do that to make sure that you have information that's known to the primary receiver that isn't lost in transmission to the secondary receiver. So that's why the name parts is important, but there is some ambiguity around use of those name parts. And I think Barry will be going into some more detail about that in the discussion points. So I think that's my last slide. I hope these examples have helped give a bit of flavour behind the guidance and help to bring our document to life a little bit. I'm going to hand over now to Barry, who's going to summarise the recommendations from the guidance and also pick out some of the key points for further discussion. Over to you, Barry.
Thank you, Sarah. Thank you, Anja. And good morning, good afternoon to everyone. So I'm going to go through a few points for discussion. You've heard the background about what we tried to do with the document and how we did the methodology, but I'm now going to look at initially some of the recommendations that come out of the document and then focus in on 3 of those recommendations in particular. So next slide, please. So there were 9 recommendations altogether. I've highlighted 3 in red on this slide, and these are the ones that I'm going to describe a bit further in a moment. So the first one is about having granular PhPIDs that are available globally, and also introduce the idea of PhPID grouping concepts and relationships. The second one is to investigate the potential benefit of the new ICSR data element that was described earlier, called imputed drug information. And not only this on its own, but also as an alternative for using name part data elements. And in the third recommendation that I'm going to focus in on, number 4 on this slide, it's around ways to improve pharmacovigilance data collection by using the information that's available via the 2D data matrix or barcode that exists already on the medicinal package. What we're looking at here is not adding new identifiers to the package, but utilizing what's already available. So there are other recommendations that are in black here, which I won't go into on this particular slide. We've already touched on having an owner for the overall ICSR guidance. If you go to the next slide, please, Christian. There's also some interest in the need for versioning with respect to the ICSR, implementing various grouping concepts, which I'll touch on for the PhPID, looking at ways of displaying contextual information and historic identifiers, as well as the basic information in pharmacovigilance drug coding tools. Looking at a wider open world view and looking at ontologies like SNOMED and how they can help in the future, and looking at requirements for medicinal product dictionaries. So I'm going to go back to the ones I highlighted in red. So the next slide, please, Christian. So first of all, the idea of the granular PhPIDs and grouping concepts. So first of all, I'm going to touch on the global nature of the PhPID because this is really important. It's one of the main benefits perceived in developing IDMP in the first place, is that if you can have a global identifier that really brings together some key concepts of the product, then this would be a real benefit, not only to pharmacovigilance, but also in other areas. But it is really important that this is a globally agreed identifier. And this is essential for consistency and interoperability, but particularly for pharmacovigilance, if you can get everyone to use a global PhPID and focus in on those three components, the substance, the strength, and the dose form, then you have the potential to collate data from a global perspective into a much larger safety database. And that will in turn give you a faster way to detect any signals that where there is something adverse happening to a particular product or substance or dose form. Ultimately, that will lead to safer medicines and improved healthcare. So that's really what we want to aim for. But global PhPID is what's really needed, not a local implementation, because that really wouldn't solve the problem. If we look at the requirements for a global PhPID, we need a number of parts of this. We first of all need agreement on the substance, and this can be done through the global substance ID. And I'm pleased to hear that there are proposals progressing on this. This has been discussed for a long time, but I think we're really making progress on this now. So this is going well. And we need a global dose form terminology. EDQM was proposed in the IDMP standard as the basis for that terminology, but there have been issues with mapping regional terminologies to EDQM, and an alternative using the IDMP dose form characteristics is being explored, and that will be part of the review of the IDMP standards that was mentioned earlier. And then global agreement on strength. It may seem relatively straightforward to express the strength of a product, but actually it's expressed in slightly different ways in different regions and between products. So we really need to be very clear when we say what a strength is. Is it 5 milligrams per mil or 50 milligrams per 10 mils, whatever it's expressed as? We need to be very clear whether it's a concentration, a presentation strength, etc. And we are making some good progress on how to do that, but there's still a lot of work to be done. And then also, if we're going to provide and maintain global PhPIDs, this really should be under a global agreement with clear governance, a clear way that we can generate those PhPIDs consistently and provide maintenance. We need a responsive way of doing that, because if you want to get those identifiers for new products or when variations of products arise, you need to have those in a reasonable time frame. And those identifiers need to be accessible. And of course, this all doesn't come for free, so somebody needs to be able to be funded to do that work. And the WHO UMC group has had a role proposed to oversee the generation of these PhPIDs, but there is still work ongoing, and there'll be a number of pilots conducted in 2022 to try and firm up on how these PhPIDs will be created and maintained. Next slide, please. So just continuing on this same recommendation, it mentions having granular PhPIDs available. So we need the appropriate PhPID granularity for the relevant use case. So for example, if you're working in a prescribing area, you may want a very precise PhPID, including the actual salt and very precise details about the strength and dose. If you're working in pharmacovigilance, as you've heard, often the data that you receive is less precise or incomplete. So you may be looking at possibly slightly less granular PhPIDs when you come to do the analysis initially. And if you go across pharmacoepidemiology, then you may need all different levels of granularity. So particularly if you come down to the substance, we've already seen an example where you could describe something as a base or a salt, but you could even go down to the specified substance level. So we need to determine what is the appropriate granularity for the PhPID and how that will be used. And then next is the grouping concepts. The IDMP standards are based scientifically on the ability to capture very detailed, thorough, and precise information during the drug development and submission processes. You want to capture as much of the information that you can in a structured form. It's already there in the dossiers and in materials that are submitted now, but we're trying to capture this in a structured form. So the PhPIDs as defined in IDMP are based on knowing specific substance or specified substance details, the exact strength and dose form. In the real world of pharmacovigilance, as we've heard, the information reported may not be that precise, or it may be incomplete. So the substance, for example, may be reported as a specific salt or a specific base, or you may get the generic base term and you either don't know what the salt is, or it may be a general term that actually doesn't really exist as a salt, but has been reported as such. Also in pharmacovigilance, you need multiple levels of analysis for the signal detection. Often you start at a high level and then drill down. So you may want to analyze data initially by grouping the bases and salts that are related to each other together, or even go up to, as was mentioned earlier, a drug class. You might not accept the drug class in the initial report, but when you come to do the analysis, you may want to go up levels. Now that isn't really part of the IDMP standard because it's looking at very precise construction of the PhPIDs. But if you can add on these additional groupings at a higher level, then that would give extra power to the analysis for pharmacovigilance. Once you've gone up to whatever level is appropriate for the initial signal detection, then you can start drilling down further into the information, going through the PhPID levels to MPIDs and PCIDs, and potentially even as far as batch IDs to get the level where you can actually detect where is this signal coming from? Is it the substance generally, or is it a specific medicinal product, or even a specific package representation, or potentially even a particular batch? So PhPID grouping concepts can be added to enhance analysis. There's nothing in IDMP that says you can't add to the standard as long as you don't conflict with the standard. And there may even be the opportunity to build upon existing concepts, for example, as used in WHODrug and SNOMED CT. So next slide, please. So this slide is just an example of where we have, on the left-hand side of the slide, we have amlodipine as a grouper concept, where amlodipine itself, as the active molecule, is never available in the real world, but it does exist in various salt forms. So you have in the screen 3 different salts which actually exist, but you have the concept of the grouper for amlodipine, which covers all of those salts. On the right-hand side, you have the same concept for ibuprofen, but in this case, ibuprofen as its base does exist, but you can still group together the base and various salts for the initial analysis. Next slide, please. Okay, so I'm now going to move on to the next topic, which is the imputed drug information field and the name parts. So if we're looking at what we've seen already in what Anja and Sarah have described, is that it's very much, the current approach is very much a a drug verbatim onwards approach. So you're starting directly from a piece of text submitted in as the drug verbatim. And what we've seen is that from that text alone, it's very difficult to go all the way down to the most detailed level in IDMP as MPIDs. So often we can identify the substance and level 1 of the PhPID. Sometimes we can get down to levels 2, 3, or 4 of the PhPID, but rarely is it possible to get down to the MPID unless we have very specific product in a particular country and it hasn't changed over time. Again, we've seen by addition of contextual information that can actually assist the coding. So the country, knowing the country narrows the list of possible products. Depending on the reporting mechanism, if you have structured entries, such as via a web form or a mobile phone app, for example, or even better from a healthcare database, then again, you can home in on the exact product and you've got a better chance of getting an MPID. We've got the timing and history aspects to see whether we need to look at older versions of the product in case for example, the MPID has changed. And we've also got the input from the primary receiver. If it's the MAH, maybe we can assume it's their own products. So these can all help us hone in on that identifier to the lowest level. And then further analysis of the drug verbatim itself can be helpful. And name parts were mentioned previously, but we didn't go into detail on that. So I just want to spend a few minutes on that. So the current ICSR and IDMP both contain data elements for structured name parts. What this means is you can dissect the product name as it is authorized into its different components. So its scientific name, its trade name, its formulation, its strength, et cetera, and you can put them into specific boxes called name parts. But there are some challenges with this, which I'll come on to in the next slide. So another option to consider is smart text searching. So if we go on to the next slide, please. So if we look at the use of structured name parts, so this is where you cut up the text and put it into specific boxes. In order for this to be effective, you need to make sure everyone has agreed and aligned on the data elements. We know exactly what they are, and I'll show you an example on the next screen in a moment. We also need to make sure that there's consistent assignment of the correct text to each name part throughout the drug life cycle. So if you start at the pharmaceutical end, where they're developing a product and are submitting information to the regulator, if they're going to put those name parts in place, they need to do that consistently. The regulators need to be able to make sure it's done correctly and approve it. And then we go through the healthcare sector and then come back through the ICSRs and pharmacovigilance. So if a drug verbatim is then received in pharmacovigilance and we want to split that drug verbatim up into its various components, we need to, again, make sure we split them up and put them into the right boxes. Then we can compare the official version that's in the label versus the data that's been received in the drug verbatim, and that can really help us home in on the actual product. Now, in the real world, we have some issues at the moment. The ICSR already describes how to do this with name parts, but it has 8 name parts, whereas the new IDMP standard has 13 name parts. Same concept, just a different number of name parts. On occasions, there's some of those name parts, it may not always be obvious where to put a particular name part. So if you have a term like forte, for example, does that mean it's a strength? Is it a formulation, or is it part of a trade name? So sometimes the assignment of the name parts can be quite resource intensive, both at the regulatory and approval end and also at the ICSR end, to actually make sure we match those name parts up correctly so that we get the right information. And if you go to the next slide, please. This slide just shows examples of the differences between the current ICSR and the IDMP standard. In the table, it's actually headed PMS because it's intended that the IDMP name parts will end up in the EMA's product management service, but it's the same set of name parts. And just one example highlighted in red, under the ICSR, you have an intended use name. So it could be things like heartburn relief or for children, but in the IDMP version, you have intended use part and target population part. So in that in the ICSR, in the IDMP version, you would actually have 2 different fields, whereas in the ICSR, there's only 1. Next slide, please. So an alternative that's been suggested is to, instead of trying to rely on the appropriate classification to name parts, maybe we could use just text searching instead. So the name parts concept was initiated many years ago, and since then, with computing power and techniques have improved significantly, and it's now we have now much more powerful text searching, natural language processing techniques, etc., that we can use. Secondly, although the name parts are included in the current ICSR in the EU version, the EU implementation guide, these have yet to be mandated. So we haven't actually used them as such. So we do have an opportunity to review options and potentially avoid resource-intensive implementation of structured name parts if we can show that having a smart text searching is a viable option. Next, please. And that just shows you where potentially the smart search could come in. So you literally search the drug verbatim and use that to impute the information without having to go through a structured name part process. Next slide, please. Okay, and then the final recommendation that I want to look at today is the use of the data matrix or straight barcode on the packaging. So currently, we've heard that a lot of the guidance that's going to be published is based on the fact that we're receiving a drug verbatim. So it's a piece of text. So it's the starting point for the process. It's step 1 in the diagram on the right. And as we've seen, this may be a free text, or it may be from a pick list or a database, so it can be a variable quality. We can apply to contextual information and maybe name parts to assist with the coding process. But even when we do that, we're usually still limited to the ability to code to a PhPID, or very rarely to an MPID, which is the goal in step 4. But we already have something on the box that we could use, and that's either the linear barcode or the 2D data matrix. Now, the current ICSR was developed before the widespread use of barcoding and imaging, so it wasn't included as part of the current ICSR. But obviously now we have widespread ability to scan and store barcodes. It's in regular use wherever you go shopping. It's on your medicinal products. It's used in the supply chain for medicinal products. It can be used in clinical settings, in hospitals, for example, and it can even be used by patients. And there are various apps on mobile phones. So scanning and photographing is something that's readily available these days. Now, the 2D data matrix on the box contains very precise information, and it also contains additional information which you can't get through the other steps 1 to 4. So if I look at what's in that 2D data matrix, you have 4 key components. You have a data carrier ID or DCID. Typically, this is a global trade identification number or GTIN, but it can be a national identifier. But you also have the batch or lot ID, the expiry date, and a unique serial number, which is used for the Falsified Medicines Directive in Europe. Now, the IDMP data model already includes a data carrier identifier data element. So if you could scan that data carrier and get that identification, then you can go straight to step 4 and you can really identify down as far as the MPID or even further than you could get to with just text analysis alone. So if you go to the next slide, please. So this is showing how it could work in practice. So if you start with the picture of the box down on the bottom middle of the screen, on there you can see in the left-hand corner of that box, there is a 2D matrix barcode. If you can scan that, then you get your 4 pieces of information, your what's called the product code, which is effectively the GTIN, the batch or lot number, the expiry date, and the serial number. They're also printed on the box as well. So this is all information that's already there. There is a data element in IDMP for the data carrier identifier. And if you go through the hierarchy of the IDMP data model, go through the package item container area, you can go up to the packaged medicinal product where you can get the PCID, which is even more detailed than the MPID. Then you can go to the MPID, which is one of the current requirements in the ICSR. From that, you can get the PhPID and the ingredient or substance ID, etc. So it gives you access potentially to the full hierarchy within IDMP. The next slide, please. Then finally, just some, on the basis of what I've been discussing, that we have some recommendations for a potential revision of the ICSR. Now, the ISO ICSR is undergoing a planned systematic review ballot. This is something that happens periodically for all ISO standards. And it's an opportunity to either suggest improvements or to vote to keep a standard as it is. This particular review closes in February next year, so we've got a bit of time to consider whether these can be put forward as part of that review. So the things I think we could put forward would be to consider the addition of the imputed drug information field. We've already seen how that could be helpful by starting with the drug verbatim and working through to the identifiers. You can also consider use of the smart text searches as an alternative to the specific name part data elements. And we know that the reason why it wasn't included originally was because many years ago, the name parts was the best way forward because text searching was very limited. And we know that name parts have yet to be used, yet to be mandated in the EU version of the ICSR. So we do have an opportunity to review that. And then lastly, if we decide that we still want to use the name parts, then the recommendation would be that we should align those ICSR data elements with those of IDMP because we need to standardize on either the 13 or the 8. It's no point having two different numbers of name parts in the two different standards. So those are the recommendations that we're putting forward. And I think that's my last slide. Next slide, please. Yeah. So I would like to stop there and turn it back to Christian to coordinate Q&A. Thank you.
Thank you very much, Barry. Thank you, Sarah and Anja before. That was a great overview of the situation regarding adverse event reporting. I remember you that, no, this is not it, that you can use the Q&A facility to ask your questions. And now I'm going to close the presentation and enable that we walk through this Q&A. So I may ask our speaker and panelists to put their camera on. And I'm going to see what is on my screen, which they also can see. But the first question is from Robert van der Stichele. Is there not a step forgotten? Before you can select an MPID, you must select the national identifier of the medicinal product or set of possible medicinal products in the country, and you will need the marketing authorization holder. And then the NCA will provide you with the MPD and the PhPID by the time it exists, and the NCA has become IDMP compliant. Who would like to respond to that? I think, Barry, that corresponds to one of the recommendations you were explaining at the end, but who wants to respond to that?
Well, certainly in the future, if you could scan the barcode, of course, that would solve the problem. If all you've got is the drug verbatim, then I think it was a case of following the recommendations we have in the document, going through the steps to try and home in on what the identifier is. And it depends how much information you have as to how precise you can get. So it's in pharmacovigilance, you only have the information that you're given or that you can clarify through the reporter. So it's sometimes limited. You don't always have all the information that you would ideally like, which is why if you could scan the barcode, effectively you would have all that information in one go. I don't know, Anja, whether you'd like to add to that from a pharmacovigilance perspective.
Yes.
No, I was just thinking about the question if I'm not sure what is meant with a national identifier of the medicinal product. Because if this is meant to be the marketing authorization itself, that national license number, that's not sufficient because we can have one license for multiple strengths, so multiple products. And then even if there would be a variation to the products, one license number will have not only multiple MPIDs, but when there are, let's say 3, but if there's a variation, these become 6 or 9 or whatever. So it's not that easy that based on a national identifier, we can easily select the MPID. Also thinking about, let's say, retrospective reporting, like Sarah mentioned, when there is something in the press getting attention and people think, oh, but I have experienced this also 3 years ago, 10 years ago. It will be very difficult to get an MPID for these type of ICSRs.
And I think also if we are dealing with national identifiers as they are used in patient care in some countries do have that national identifier and some countries don't. Some countries it applies, the scope is prescribed products, but of course ICSRs can happen to a whole range of products, including over-the-counter products that we might not even consider medicines, but the patient does, and may indeed have interactions and all sorts of things. So there is definitely a need, if you have got national clinical identifiers like the Z index that Anja mentioned, or the NHS DMD in the UK, to have that mapping between them. But we're not there yet. And even if we do have that, there will be things that will be outside of that.
But may I ask you just to react on this? If you read the question a bit differently, the national identifier would be this data matrix. What would be your response? You read the data matrix?
Yeah, I mean, if you can read the data matrix, then you can get a link directly into the whole hierarchy of IDMP identifiers. So you can get everything you want, right down to PCID, even batch ID. So it's even more precise than going through the drug verbatim route. So that's the ideal way to do it, if you can do it. But that's the future. We're not there yet, as you'll probably hear a few times.
But,
and that's fine if we're looking at a dispensed medicine, but if all we have is information about the prescription, which quite often we have, if it's not coming from the patient, it's coming from the clinician, then we won't have a DCID, we'll have something much more generic, which many of the national drug dictionaries have, but by no means all.
But you have just to look at the time when the question was raised. It was shortly after 3:30. So it was before Barry explains the recommendation also. That's the reason why I insisted it. I suggest we go to the next question. Jean-Gonzague Fontaine, do I have correctly understood as marketing authorization holder, when the product is not well defined, it should then be reported on the PhPID of the marketing authorization holder. So adverse events on heparin A vaccine, or whatever it is, will be reported on Havix 720, Havix 1440 by GSK, etc., by Sanofi and by MSD, and something else by WHO. Please note that even the PhPID level 1 will not be the same for all these products. The substances are not the same, biologicals. Wouldn't it be easier to define a kind of a generic PhPID, in this case, derived from the ATC? Wow, that is a question. Who wants to respond to that?
Well, I think one of the principles that was explained in the slides was that you can use the if the report goes to the marketing authorization holder, then that marketing authorization holder maybe can assume that it is their product for the purposes of processing, and therefore, if it goes to GSK, it would be one of those Havrix options. So you'd code it to the closest available PhPID. for that particular product. Yes, if it was a different company, it would be a different vaccine and therefore a different substance, etc. But I think the responsibility is with the MAH to report it, assuming it was their own product. Now, where the grouping might come in handy is if the collaborating centres, the regulators look and collate their data and look at it and say, well, I have these reports from GSK, I have these reports from Sanofi. Actually, if I group them up and say, what about all COVID vaccines, or what about all this type of vaccine? Do I still get a signal, or is it specific to GSK or Sanofi? And I think that's where the grouper concept comes in, but at an even higher level than I tried to explain. And then, I mean, they maybe not be PhPIDs, they may be something else, but it's the wider grouper concept. It's actually one of those other recommendations in the document.
Thank you. Another comment? By the way, the name document has been mentioned several times for the participants. After this webinar, you will receive an email from Zoom, and in that email you have a hyperlink to the drafts documents, which, as the caveats mentioned by Anja van Haren, is not something official at all, but it gives you access to the information. Good. I continue with the next question. Is there a connection to the reason why medicines are prescribed? If so, how? For instance, with ICD-SNOMED, could there be a standard and understandable directive for patients to report also on how the working can be registered, like more or less pain, possibly with ICF? Who wants to address this challenge?
I can pick it up a little bit, but I think obviously the closer to the patient and the person who is and the record which holds the information, the closer you can get to move the data from, the better and more accurate the reporting is going to be. So that might be SNOMED, it might be ICD, depending on what is being used in the systems. There's a lot of very similar information in SNOMED as there is in ICF, but I'm not sure of where ICF is being used in the systems which would link up to ICSR and adverse event reporting. But there is nothing to say that you couldn't use ICF to be to actually record the impact on an individual, nor why you're giving it in the first place, and any changes that are happening as a result of a drug. But it would be about what's actually in the systems, because that's the whole point of this, is getting that end-to-end flow of data, and actually how we create it properly to enable data movement.
Thank you. Other comments?
I was just thinking that perhaps it's also good to clarify that in the ICSR there is a data element to provide the indication for the product, and this is not automatically taken from the drug information, also because there can be off-label use, misuse, medication errors, things like that. So it's not linked automatically.
But that's where we're going, isn't it? That's where we want to start to move. So, yeah.
Thank you. Next question, rather technical, I think. On the slide related to invented substance, the correct strength or expression of molecular weight and therefore varies. In the invented example, technically the strength does not exist. There is no chemical form. How would strength then be handled?
I'm trying to find which slide that refers to.
Maybe I can make a general comment. I mean, this is one of the challenges we have currently with defining the PhPID, as to how do you express strength and do it consistently across every region. And there are many examples where there are different ways to express strength that I know WHO, UMC, and others are trying to come up with a standard way of handling that. So it's part of a general issue.
Maybe if Julie, you are searching still. Okay, then once you search, we go to the next question because the time is passing. Maybe it's a simpler question. Will the DCID be part of the PMS data delivered by the marketing authorization holder to the NCA?
Maybe I can have a go at this one as well.
Yeah.
Okay, so in the IDMP model, as I said, there is a DCID data element, and that's also part of the PMS data model. And the final version of the EU implementation guide isn't ready yet. I don't know when that will be finalized, but there will be instructions on there that you know, this field should be filled in as part of the PMS data. How and when that will be filled in is still to be determined, but the intention is that that will be there. So in theory, it should be available to all the NCAs.
Thank you. It's a good hope. Julie, do you have the answer for the or comments to this question you had?
I can't find the exact slide. Apologies.
Oh.
I think, as Barry was saying, one of the challenges that is still under discussion and looking at for resolution is around the fact that we can describe strengths differently. You cannot divorce the way that you describe the substance from the way that you describe the strength, which I think actually is possibly at the heart of David's question. And even particularly in our verbatims, we don't always get that very strict tie-up that we might as scientists want to have. Patients, particularly when they're reporting in verbatims, don't have that. So we have to work through all of those challenges, some of which IDMP can help us with, because it allows us to see the different expressions of strength and how they relate to the different substances, and some of that which IDMP doesn't help us with, because patients are putting different things together and we have to interpret the verbatims.
Thank you. We have a last question. I think it is an interesting one because it reflects also some thoughts we have in the context of UNICOM and elsewhere. Our colleague says, I like the concept of imputed drug information. Do I understand well that this is a structured set of characteristics of the drug that is appended to the ICSR? Would those be identifiers or attributes? And then he has another comment to explain about identifier and attributes. He says the issue we're just relying on identifier is that we lose information by rounding up to the nearest identifier, or we just won't ever have enough identifier. So the main question is it a structured set of characteristics of the drug appended to ICSR with identifier or attributes?
It's more around the context of the ICSR itself, helping us to identify which is the medicinal product that is having the finger pointed at in the adverse event submission. I think, as we tried to show, sometimes we can't get the exact detail from the verbatim. The reporter hasn't said, and it was this brand with this strength with this manufacturer, but they have said, we do know that it's this country and this brand name, and we know that there is only one strength and one manufacturer, so we can impute. So the imputation information is coming from the context of the report. It's not so much about a structured set of characteristics of the drug itself. And if my pharmacovigilance colleagues could add to that, I'd be very grateful.
Thanks,
I would
Julie.
see it as a sort of a modified verbatim that contains more information that then you can then use to match to the correct term in the database. But I'll hand over to Anja.
No, I think both of you explained very well. The idea is that just another free text data element is added to the drug sections of the ICSR to, as Barry also nicely phrased it, as to capture the modified verbatim. Also thinking of the example that Sarah gave, where the verbatim was a COVID vaccine, but from the context you know it was Spikevax. Currently, there is no data element in the ICSR drug section to capture this modified verbatim as Spikevax. So this would be information that would now be lost if we only have the strict drug verbatim in the ICSR. So it's just a proposal to add another data element.
But it would also make that link, wouldn't it, between the drug verbatim and what you're coding as the term identifier. And otherwise, the secondary receiver might not understand why you've gone from that particular verbatim to the particular term that you've selected if you're not showing somewhere in the ICSR what the imputed information is. So that's sort of the idea behind that free text option, as Anja said.
Thank you. I think that was a very good explanation to have a better understanding of what is expected in this new field. We have a comment in the chat box about the question we had before regarding the use of ICF. But I leave, I suppose, because of the time, I leave that response just to your knowledge. Oops, sorry. And I want to, just before closing, to make an announcement. I suppose everybody knows we are narrowing the end of the year. That's something not new, really. But what is new is that our next community of expertise will be on the 14th of January. And it will be on the theme of the vaccine challenges, cleansing, confidentiality, and vaccine naming. And I think this is also going to be highly interesting. We have appreciated that we had a large number of registration for today, and we hope that we continue on that curve, that we can reach out and, share the knowledge and the information with the communities on that way. Again, also, you have in the meantime this satisfaction questionnaire, which we invite you to answer. That's highly important to us. With that, with this, sorry, I want to thank speakers and panelists for this really living session. This time we had really the interaction. We had 1 hour very dense on the, explaining what we want to share, and half an hour to share that and just debate that with the audience. I think that was the best way possible to have an interactive community of expertise. So thank you to speaker and panelists for their help to achieve this. With that, so I close the session. Again, don't forget the questionnaire. And when you receive the Zoom email, it will be on Sunday. You also find the hyperlink to find this deliverable from UNICOM, as we mentioned before. Now, thank you. Have a good rest of the week and an excellent weekend, and keep safe. Thank you and goodbye.
Thank you. Goodbye.
Bye-bye.